Today we will discuss about the phase contrast microscopy. Phase contrast microscopy is a special type of compound light microscopy having some advantages. With the help of phase contrast microscopy, we are able to see the living and unstained specimen. In case of conventional light microscopy, unstained specimens such as cells present a unique problem for light microscopists because the images generate very little contrast and are essentially invisible in ordinary light microscopy. This is even true for transparent periodic specimens such as diatoms. Although transparent objects induce phase shift to interact beams of light due to scattering and diffraction, they remain nearly invisible because the eye cannot detect differences in phase. The phase contrast microscope, which transforms differences in the relative phase of object waves to amplitude differences in the image. In the 1930s, Frits Zarnik, a Dutch physicist at the University of Groningen in the Netherlands, created an optical design that could transform differences in phase to differences in amplitude. The development of phase contrast optics is a brilliant example of how basic research in theoretical optics led to a practical solution for viewing unstained transparent objects in the light microscope. The Zeiss optical works in Jena introduced phase contrast objectives and accessories in 1942, which transformed the research of biology and medicine. For this excellent invention and theory of image formation, Frits Zarnik was awarded with Nobel Prize in Physics in 1953. Now, phase shift. Please see, at the top of the diagram, there are three wavelengths of light. The first one is having the amplitude of A1 and second amplitude A2, but both the light is having the same crest and trough of the light wavelength, and both the wavelengths are the same. So the light of that type of amplitude and wavelength may form positive interference that will result the addition of both the light of same wavelength but having the different amplitude. The final amplitude will be A1 plus A2. That will be the greater amplitude. Amplitude of the light wavelength determines the brightness of the light. If both the light wavelength having different amplitude but they are in phase, then the positive interference will be created and the higher amplitude will result the brighter image and brighter light. But the diagram in the bottom, both are having the amplitude of A1, A2, but their crest and trough are not in phase. So they will create a destructive interference and the final amplitude will be resulted A1 minus A2. That will be the shorter amplitude of light wavelength, and that will produce a dull light, and dull light will create a dull image. This is the optical elements of the phase contrast microscope. In phase contrast microscope, there are two special optical instruments are used, that is phase plate and phase annulus. Phase plate is placed inside the objective lens and phase annulus is placed above the condenser, but both the phase plate and phase annulus are complementary to each other. Here in the diagram, the light rays are passing through the condenser, then through the phase annulus and passing through the specimen, then entering into the objective and then passing through the phase plate. Here in the phase contrast microscope, the advantage is that in case of biological specimen, it changes the shift of phase, that is lambda by 4, and this lambda by 4 changes of the biological specimen is not perceptible to our eyes, but due to the application of the phase plate and phase annulus, an addition of lambda by 4 phase change will be created. So the final phase change will be lambda by 4 plus lambda by 4, that is lambda by 2, and this change will be perceptible to our eyes. In the next diagram, you will clearly understand. Here phase annulus and phase plate are shown. The left side light rays are passing through phase annulus and missing the specimen. So there is no change in phase, because biological specimen changes the phase up to lambda by 4, but here the specimen, the light is missing the specimen. So the phase change is not occurred here, and it is also passing through the thinner portion of the phase plate. So there will be no change in the phase shift, and phase shift is not occurred here. In case of middle rays, the light ray is passing through the phase annulus, then passing through the specimen. Due to passing through the specimen, there is a change in shift of light, that is lambda by 4, but it is also passing through the thinner portion of the phase plate. So the final phase change will be lambda by 4, and this is also not perceptible to our eyes. But in case of right side diagram, right side light ray, in that case, due to passing through the specimen and slide, there is a refraction of the light. The light ray is passing through the specimen, then there is a change of phase shift, that is lambda by 4, and then it refracts, and after refraction it bends and passes through the thicker portion of the phase plate. So there is another additional phase change of lambda by 4. So the final phase shift will be lambda by 4 plus lambda by 4, that is lambda by 2, and this phase change is perceptible to our eyes, and the greater the phase shift, that is transformed into amplitude, that means the greater amplitude will create a brighter light, and brighter light will create a brighter image. This is the advantage of phase contrast microscopy for the use of and for the conversion of changes of phase up to lambda by 2, that creates a greater image, and unstained specimen can be seen with the help of phase contrast microscope. And the main application of the phase contrast microscope is to see the basic structure of the microorganisms and cells without staining, as well as we can visualize the cellular motility, as well as we can visualize the cell division, that is the binary fission in case of bacteria. It is seen very easily with the help of phase contrast microscope. Staining is not needed here because before staining, the cell fixation of the cell should be needed, and fixation of cell can be done either by heat fixing or by chemical fixing, and both are hazardous to the cell, and this type of fixing results the cell death, and the stain, which is a hazardous chemical, may also alter the shape of the cell. So we will not be able to see the cell in their exact condition as well as in the living condition. So to overcome this type of problem, phase contrast microscope is used. And here some diagrams are also shown, and these diagrams are taken from the microbiology book by Prescott. Here you can see that these cells or these microorganisms are in their natural color. No stain is applied. Here the endospore of Clostridium botulinum is shown. The structure of Paramecium is also shown, and S. volutans are also shown. That's all. Thank you very much. Thanks for your patience watching.